Good morning dear students and welcome to Genesis. Well, this is my last video on the chapter photosynthesis और आज के इस lecture में I will mainly cover two experiments. One, to prove that carbon dioxide is necessary for photosynthesis, and the second experiment is to prove that oxygen is given out during photosynthesis. और last में हम थोड़ा सा समझेंगे importance of photosynthesis. Well, to start this experiment, this is the experimental setup where what do you need? You need a potted plant, and इस potted plant को आप destarch करेंगे like we do for other experiments, like we keep it out for a stipulated period of time. कितने घंटे के लिए बाहर रखना है वो आप मुझे comment box में लिखिएगा, because by now you should know this because मैंने मेरे सारे videos में ये explain किया हुआ है. Now then you take this out in the sun, but before you take this out in the sun आपको एक experimental setup बनाना होगा where you are going to keep a conical flask attached to half portion of the leaf which is still with the parent plant, and this conical flask is not empty, it has a chemical inside. What is the name of the chemical? It is potassium hydroxide. Please remember the name. Why is this chemical important? Because it absorbs carbon dioxide. Very very important. मतलब wherever this chemical is present, that particular container will contain no carbon dioxide because it will be absorbed by potassium hydroxide. Done? Now you keep the setup out in the sun for some time, then you pluck the leaf and perform starch test. Now before I explain what will happen in the starch test, let's see this leaf. So once you pluck the leaf, what do you expect? This is the part of the leaf which was open or which was exposed to the atmosphere, and this is the first part which was in the flask. So indirectly I can say this is the part where there was no carbon dioxide, and this is the part which could access carbon dioxide. Hence in this part photosynthesis will take place in the region A, but in this region B there will be no photosynthesis as there was no carbon dioxide. So now when we perform starch test on this leaf, we are going to see this part which is part A will give us positive starch test. And इसका मतलब क्या है? The iodine will change color from brown to blue black, whereas part B will give us negative starch test. ऐसा क्यों? Because it had no carbon dioxide, so did not perform photosynthesis, could not produce starch. As a result of which, जब हम यहाँ iodine डालते हैं, then the iodine retains its color. That means it remains brown in color, does not get converted to blue black. So this is the entire experiment that can easily prove that in absence of carbon dioxide, there will be no photosynthesis. If there is no photosynthesis, no starch production, and this can be very easily proved by a basic starch test on the leaf. Now just remember the name of this chemical, because आपको exam में पूछा जाएगा name the chemical which is used to prove carbon dioxide is necessary for photosynthesis. Your answer will be potassium hydroxide. Now let's quickly do the next experiment. To prove that oxygen is given out during photosynthesis, but for this experiment we are taking aquatic plant hydrilla. One question that can come to your mind is, why are we taking aquatic plant? Why not a terrestrial plant? In a terrestrial plant, when they perform photosynthesis and when they release oxygen, it is very very difficult for us to find that out, right? Because the gas immediately mixes in the air and we cannot see it. But if the same thing is done by an aquatic plant, then the oxygen gas which is given out can be easily understood or can be seen by the number of bubbles that come out from the surface of the leaves. So that is why we have wisely chosen aquatic plant for this experiment, because with terrestrial plant it will be very difficult for us to understand when the oxygen gas is getting released. So what do? We take a beaker, and in this beaker we take aquatic plant hydrilla, and we fill the beaker with water, preferably pond water. You also have to assume one thing, that this beaker this funnel should remain in place with the help of two supports, any support, because otherwise the funnel will have a tendency to go up. So what did we do? We took hydrilla, we have covered the hydrilla with a funnel, and then we have filled the beaker with water. Now when you're filling the beaker with water, you just have to remember one thing: the level of the water should be above the stem of the funnel. That is very necessary. Okay, so the level of the water has to be above the stem of the funnel. You keep this experiment and then take a test tube filled with water, invert the test tube in such a way that it completely fits into the stem of the funnel, and now you keep the setup as it is out in the sun for some time. What are you doing is, you are here, remember one thing, there is no concept of de-starching because you are not going to test the presence of starch. What you just want to see is whether the plant releases oxygen during photosynthesis or not. So now you keep the setup as it is out in the sun for some time. What you are going to observe is there are small bubbles that are going to come out from the stem of the funnel, and then they will start to accumulate in the test tube, and the level of the water in the test tube will eventually be pushed down by the accumulation of gas here at the tip of the test tube. So you're going to see that something is getting collected, and that is causing the level of the water in the test tube to go down with time. But which gas is this? We have no idea, but we know some gas is definitely getting collected. Now we have to test and find out if this gas is oxygen or not. For this, the next step, which is very, very tricky, what you have to do is you have to take out the test tube and seal the mouth of the test tube with a cork, and everything that I said will be done underwater. Not that you can take out the test tube completely from the beaker and then seal the mouth with a cork. No, you have to do it underwater. Why are you doing this? Because you do not want any other gas to get mixed in this. Okay, so as soon as it comes out, you are going to, as soon as you take out the test tube, seal the mouth with a cork underwater and then keep the test tube outside. What you're going to do is you're going to bring a splinter, a burning splinter, close to the mouth of the test tube. So you open the cork and bring the splinter close, or insert it inside the test tube. The moment you do this, you're going to see the flame, which was initially this low, suddenly increases as it enters into the test tube. So there must be some gas which is supporting the burning of the splinter, and we know in nature there's only one gas which supports the burning, and that is oxygen. So now it is evident that yes, of course, like we already know, during the chemical reaction of photosynthesis, we mentioned that oxygen gas is evolved, but now we also proved that yes, oxygen gas is given out during the process of photosynthesis. So in the initial step, you only get to see bubbles getting collected, and these bubbles they come out, and there is collection of some gas up here in the tip of the test tube. To prove that this is oxygen, you need to bring a burning splinter close to the test tube, and if it burns with a flash, then definitely that gas is oxygen. One more thing, this rate with which the bubbles are getting released can be increased. How? You can just add some sodium bicarbonate into the water, and you see that the rate with which the bubbles are getting produced will increase. Why? Because this chemical increases the amount of carbon dioxide dissolved in the water, which will be readily utilized by the plant to perform photosynthesis, and since the rate of photosynthesis will increase, the rate of evolution of or emission of the gas will also increase, and so the rate with which the bubbles are released will also increase in the process. Now let us understand the importance of photosynthesis. Now फोटोसिंथेसिस क्यों इम्पॉर्टेंट है ये अभी तक तो आपको समझ में आ ही गया होगा. देयर आर टू मेन रीजंस टू इट. नंबर वन, दिस इज द प्रोसेस बाय विच फूड गेट्स प्रिपेयर्ड फॉर ऑलमोस्ट ऑल लिविंग ऑर्गेनिजम्स दैट वी सी अराउंड अस. ये मैं कैसे एक्सप्लेन कर सकती हूँ? फॉर एग्जाम्पल, दिस इज द प्रोसेस बाय विच इनऑर्गेनिक कार्बन डाइऑक्साइड एंड वाटर कैन गेट कन्वर्टेड इनटू ग्लूकोज, राइट, विच इज द सिंपलेस्ट फॉर्म ऑफ शुगर, जिसे हर ऑर्गेनिज्म बहुत आसानी से डाइजेस्ट करके उससे एनर्जी रिलीज कर सकता है. नाउ because of this we always keep plants as producer. हम इन्हें producer कहते हैं. Why producer? Because they are kind of producing food for the entire living community. अब इसी प्लांट से कोई भी प्लांट हो सकता है, सोचो उसे कंज्यूम किया किसी ग्रास हॉपर ने, एंड इस ग्रास हॉपर को कंज्यूम किया एक रैट ने, और उस रैट को कंज्यूम किया एक हॉक ने. सो व्हाट हैव आई रिटन? आई हैव रिटन अ फूड चेन इन फ्रंट ऑफ यू वेयर आई एम ट्राइंग टू एक्सप्लेन यहाँ कौन कंज्यूमर है एंड कौन प्रोड्यूस है. तो प्रोड्यूसर तो एक ही है, which is the plant, but ये तीनों consumers हैं. The grasshopper is a consumer, the rat is also a consumer, and the hawk is also consumer. Difference बस ये है, grasshopper is a primary consumer. क्यों? Because ये directly प्रोड्यूसर से फूड ऑब्टेन करता है एंड ये सेकेंडरी कंज्यूमर्स बन जाते हैं. सो द रैट हियर इज द सेकेंडरी कंज्यूमर एंड द हॉक हियर इज द टर्शियरी कंज्यूमर. सो अब हम लोग को पता चल गया कि what is the difference between a producer and a consumer. The one who can convert inorganic substance into organic substance and make food for itself as well as for others, वो हो गए producers. वो सिर्फ plants हो सकते हैं और rest जितने भी organism है वो consumer हो जा रहे हैं because we are directly or indirectly dependent upon the plants. Next very important thing that is produced during this process is oxygen. We all know this is the process by which oxygen gas gets liberated and oxygen gas is necessary for all the living organisms to produce energy. अब कोई भी खाना जो खाते हैं उसको break down करके energy release करने के लिए oxygen की ज़रूरत होती है. So if there is no oxygen we will not survive. There is another reason why oxygen is also very important. It is to support burning. हम सबको पता है this is the gas that supports burning. So these are the benefits of or importance of photosynthesis. One, it produces food and that is why it is known as producer, and second, it produces oxygen which is essential for almost all the living organisms. So with this we finish this chapter. अब हम लोग next chapter की तैयारी शुरू करने वाले हैं. I hope you are revising because अगर आप नहीं revise करते हैं it is useless. Biology revision based एक subject है. Revise more and more. And we are going to meet soon in the next video, and do subscribe my channel and do share it with your friends. It's really going to help me in growing my channel. Thank you.